Hi Sir John, my name is aurelia gunawan from 12 IPA B. So in this video, I'm gonna explain about the effect of tea on amylase. So this material and tools we need. So they're white tea, beaker glass, syringe, test tube, and small beaker glass. So we need thermometer and hot water. Next, we need iodine solution, black tea, amylase, and also starch. So first, label 3 test tubes S1, S2, and S3. And label another 3 test tube with A, AT, and T. Here I number the white tile, number 1 until 10, and also A, AT, and T. So here use a syringe to put 2 cm of starch into S1, S2, and S3 tubes. Here I also put 2 cm of amylase solution into A and AT tubes. So here I'm using a clean syringe to put 2 cm of T into AT and T test tube. So after all done, we need hot water to be added to the beaker glass. Then we have to wait until 40° hot. So the next step, place all 6 test tubes into the hot water bath. Then we have to wait for 2 until 3 minutes. While waiting, add 1 drop of iodine solution to each hole. For now, it's section A first. After 2 minutes, pour the starch in the S1 test tube into A test tube. Then mix it with a clean syringe. Use a pipette to immediately remove a drop of the mixture from test tube A and place it onto the first drop of iodine solution in section A on the white tea lay. Repeat every 30 seconds until all 10 drops of iodine solution in section A have been used. Reset the stop clock to 0. So now pour the starch in S2 test tube into the AT test tube. And mix with the clean syringe. So now pour 1 drop of iodine solution into each hole of AT section. So now use a pipette to drop off the mixture from AT test tube and place it onto the first drop of iodine solution in section AT of the white tea lay. Repeat every 30 seconds also until 10 drops of iodine solution in section AT have been used. Also, reset the timer to 0 for each drop. Okay, same step, we have to drop 1 drop of iodine solution into each hole. For now, it's section T. Okay, now pour the starch in S3 test tube into T test tube. And mix it with a clean syringe. Immediately drop off the mixture from test tube T and place it onto the first drop of iodine solution in section T of the white tea lay. Repeat every 30 seconds until all 10 drops of iodine solution in section T have been used. Don't forget to reset the timer to 0. And yeah, this is my experiment result. So this is my hypothesis. Tea contains tannins, which are known to inhibit the activities of enzymes such as amylase. If tea is added to the mixture of amylase and starch, then the amylase will work more slowly in breaking down starch into the simple sugar. As a result, the starch will take a longer time to disappear and the iodine taste will stay dark blue for a longer period compared to the mixture without tea. This is because tannins can bind to the amylase enzyme and change its structure, reducing its ability to catalyze the breakdown of starch. So here are the conclusions. In this experiment, we test that the effect of tea on the amylase by using iodine on a white tea leaf. So first, starch was mixed with amylase and every minute a sample was dropped onto iodine. If starch was still present, the color turned dark blue or black. If the starch has been broken down into simple sugar, the color changed to light brown. From the result, we saw that in the tube that without tea, the color quickly turned into light brown, showing that amylase worked properly to break down the starch. However, in the tube that with tea, the color stayed blue or black or dark green, meaning the amylase works slower. This happened because tea contained tannin, a type of polyphenol which can bind to amylase and change its active site, making it more harder for amylase to break down the starch into sugar. So that's it for my explanation. I'm Aurelia Gunawan signing out. Thank you, Sir John.